In this video, we're going to start our conversation on the cardiovascular system. We are going to focus primarily on learning about blood typing, and hemoglobin. This information can also be found in Chapter 17 of your OpenStax text. So in this first video, we are going to describe the cardiovascular system and its role in transporting substances and maintaining homeostasis in the body. We're going to describe the functions, composition, and physical characteristics of whole blood and explain why it is classified as a connective tissue. We're going to identify the components of plasma and explain their roles in maintaining blood function and homeostasis. And then finally, we're going to explain the structure, function, and life cycle of erythrocytes, which are red blood cells, including the chemical composition and role of hemoglobin. All right, so when we think about the cardiovascular system and its role in kind of maintaining homeostasis within the body, it carries out a couple of different functions that helps it do and carry out the work that it does. And so it plays an important role in the transportation of substances throughout the body. So it's going to be very important for the movement of oxygen and carbon dioxide to and from our tissues. When we think about the role it plays with the digestive system, there's going to be nutrients and other molecules that can be picked up in our blood vessels and carried to various parts of the body. And then when we think about the excretory function, it can carry metabolic waste to kidneys to be excreted from the body. It's also going to play a role in regulation of our fluids and body temperature. And so one of the ways that it kind of helps with temperature control is it has the ability to divert or and move blood to the skin or away from the skin in a way to cool or warm the body. So what you'll learn are terms vasodilation and vasoconstriction, where we are either expanding the blood vessel or making the blood vessel smaller. And in doing that, we can kind of divert blood to and from the surface of the skin or surface of the body rather to maintain temperature. Okay. It also plays a role with protection. There's cells that can, come together and plug breaches and abrasions and cuts that may occur to the skin in a process called blood clotting or hemostasis. We also have the immune function, where we have specialized cells that can fight against pathogens and invaders. Our leukocytes or cytokines can all work together to fight against foreign invaders. When we think about the components of the cardiovascular system, it's going to include blood, it's going to include our blood vessels, and the primary machinery that kind of moves the blood and pumps the blood out and receives the blood from the body is going to be the heart. All right, so let's talk about blood. So blood is a fluid connective tissue. When we look at the texture, it is sticky. When we look at its color, it is slightly opaque with a metallic taste. The color is going to vary with the concentration of oxygen that is present within it. The pH of blood is maintained within very tight parameters. So you're going to expect blood to have a pH of anywhere between 7.35 and 7.45. Blood is going to represent anywhere from 5 to 8% of one's body weight, and that's going to vary between males and females. Okay, so overall, we're expecting between 4 to 8 liters of blood contained within the chambers of the heart and in our blood vessels. But on average, in males, they're going to have about 5 to 6 liters, and in females, 4 to 5 liters. All right. So let's look at the parts of blood. Blood is going to be made up of plasma, which is this yellowish fluid that makes up about 55% of our whole blood. Okay, plasma is going to contain primarily water, but also proteins and other solutes like electrolytes, nutrients, respiratory gases, and waste products. We also will see a concentration of what is called formed elements. And formed elements is just a fancy term for cells. So these are the cells that are going to make up, that are going to be found within our blood. Okay. Now separating the plasma from the formed elements, you're going to see this buffy coat. And a buffy coat is just a thin layer of white blood cells and platelets in a blood sample that has been separated using a centrifuge. And a centrifuge is one of those little machines that you stick your test tube in and you hit start and it like just shakes it and mixes it. Okay, so when we mix whole blood, we can separate it into its counterparts. Otherwise, it's all going to just kind of look the same color. Okay. The hematocrit is going to show the proportion of red blood cells in the blood, which helps us assess the body's ability to transport oxygen. Okay, so normal values for male is a hematocrit of about 47% plus or minus 5%. In females, about 42% plus or minus 5%. The CBC, or the complete blood count, is a common test that we use to evaluate the levels of different blood cells. So we can figure out the hematocrit, how many red blood cells are present within the whole blood sample, how many white blood cells are present within the whole blood sample, platelets, etc. So it just helps doctors be able to diagnose conditions like anemia or infections or any blood-related disorders. Now, serum is the clear yellowish liquid portion of blood that remains after blood clots. It's going to contain water, electrolytes, proteins, hormones, and waste products, and it's going to play a key role in transporting nutrients and supporting immune function. Now, because this is a particular part of the blood that has been removed and does not contain fibrinogen, it is not going to clot. Okay, so again, it is the fluid that is left over after blood clots. Okay. Plasma volume refers to the volume of the liquid component of blood known as plasma, which makes up about 55% of total blood volume. So let's take a look at plasma. Alright, so plasma is this straw-colored sticky fluid made up of mainly water, roughly 92% water, containing over 100 dissolved solutes that are going to include nutrients, gases, hormones, waste products, proteins, and inorganic ions. Plasma proteins are going to be the most abundant of the solutes present within plasma. And plasma proteins serve a variety of functions, but they are not normally taken up by cells to be used as fuels or metabolic nutrients. Instead, they're going to remain in the blood, and most of the plasma proteins are going to be produced by the liver. Okay. Albumin makes up 60% of our plasma proteins, and it serves as a carrier molecule in the blood. It maintains the blood osmotic pressure, and albumin has amino acids that have functional groups that can donate positive and negative charges, and so in that sense, it also serves as a buffer. So the formed elements in blood are just the cells that make up blood. Formed elements are going to include white blood cells, red blood cells, and platelets. Only white blood cells are complete because when we look at the components of a white blood cell, they're going to feature the structures that we would find in a cell, whereas red blood cells and obviously platelets, they don't have nuclei. Or other organelles. They don't have nuclei, they don't have mitochondria. They are simply designed to to grab onto oxygen or carbon dioxide and shuffle them to the places within the body that needs it. Okay. Platelets are cell fragments, so they're not going to have any kind of machinery or structures within them as well. Most survive in the bloodstream for only a few days. Now, most blood cells are going to originate in the bone marrow, and they do not divide, so they do not undergo the process of mitosis. All right, so we're going to talk about red blood cells. The anatomical term for red blood cells is the erythrocyte. Red blood cells are fairly small, roughly about 7.5 micrometers. They resemble these flattened biconcave discs, and they lack nuclei and mitochondria. And so because they don't have a nucleus, we call them anucleate. A meaning without, and then nucleate referring to the nucleus. They have a half-life of about 120 days. And when you look at these under the microscope, because they do lack some of those organelles and those structures, you're going to notice these little pink reddish circles on the tissue sample that sometimes have an area in the middle of the cell that just looks like a whitened area, like there's nothing there because there isn't anything there. So red blood cells contain hemoglobin, and hemoglobin is a protein that allows the cells to carry oxygen and carbon dioxide. Each red blood cell has the ability to carry 280 million hemoglobin, which I'm going to talk about that significance in just a bit, and its ability to be very efficient in oxygen delivery to our tissues. Now, red blood cells are removed by a process called phagocytosis, where macrophages in the spleen and in the liver can engulf and break down old or damaged red blood cells. The hemoglobin that is broken down into its components, and parts of it may be recycled and other parts of it may be removed from the body. Now, some of the features that make erythrocytes efficient for gas transport, the biconcave shape gives it a huge surface area relative to the volume for gas exchange. Hemoglobin makes up about 97% of the cell's volume, not including water, so the majority of the red blood cell is going to be saturated with these hemoglobin molecules that can bind to oxygen to impact and increase the efficiency of the transport of that bloody gas. Red blood cells have no mitochondria, so all of their ATP production is anaerobic. They do not consume the oxygen that they transport, okay? Which is very important, because if they did, then that would just reduce the amount of oxygen that could be delivered to the tissues. So hemoglobin binds reversibly with oxygen, and what that means is that hemoglobin can easily pick up oxygen and easily let it go. And so hemoglobin will pick up oxygen in the lungs. Oxygen is going to bind to the iron in the hemoglobin to form oxyhemoglobin, and then release it into the tissues where the oxygen is needed. This reversible binding allows hemoglobin to efficiently transport oxygen throughout the body and adjust to different conditions. Now, when we look at the components of hemoglobin, hemoglobin consists of a red heme pigment, which gives it the reddish color the reddish color that we see, bound to the protein globin. Now, globin is going to be composed of 4 polypeptide chains. There's going to be 2 alpha chains and 2 beta chains, as reflected on your screen. A heme pigment is bonded to each globin chain, and within each heme pigment, you're going to see an iron atom that has the ability to bind to 1 oxygen molecule. Now, each hemoglobin molecule can transport 4 oxygen, and each red blood cell contains roughly 280 million hemoglobin molecules. And so if we take that number, and I'm going to do it right here on my phone, I may be off a little bit, but we'll see if my phone will do numbers this big. We're going to multiply 280 million by 4, and we get 1.12 billion oxygen molecules can be contained on 1 red blood cell. That is a lot. This incredible oxygen carrying capacity is what allows your blood to efficiently transport oxygen from the lungs to the tissues throughout the body. Now we have a couple of terminology that we need to go through. When our red blood cells pick up oxygen in the lungs, we call that oxygen loaded in the lungs, and it's going to produce oxyhemoglobin. Okay, and this is going to be why arterial blood looks very, very bright red, okay, because it is oxygen rich. Now, at the level of the tissues, the red blood cells are going to unload oxygen at the tissues. It is going to produce what is called deoxyhemoglobin. It just means that there is a reduced oxygen bound to hemoglobin, producing that darker red color that we see. Now, at the level of the tissues, when we are unloading oxygen, we're going to be loading carbon dioxide onto the hemoglobin molecules to bring carbon dioxide back to the lungs to be removed from the body. So 20% of the carbon dioxide in the blood is going to bind to hemoglobin, producing what is called carbaminohemoglobin. Okay, carbaminohemoglobin. Now, I will say that this is a question, not to be a trick question, but this is a question I typically put on the exam where I'm asking you about these terms specifically, okay? And one term that typically gets thrown into the multiple choice question is carboxyhemoglobin. That is not something that we've discussed, and that is not something that has been defined here. So be very careful and make sure that you organize your notes so that you have the definitions of the terms we have discussed on this slide, okay? Blood cells are created from a process called hematopoiesis. And blood cell creation is always going, or I won't say blood cell creation, but all of our cells are always going to originate from a stem cell. So when we talk about the production of blood cells, it's going to be some type of hematopoietic stem cell, okay, that will differentiate into another stem cell. In this particular case, it is the hemocytoblast. And that will differentiate into either a myeloid stem cell, which is cells that originate in the bone marrow of our long bones, or a lymphoid stem cell, which is just cells that are going to be present in our lymphoid tissue. There are two types of hematopoiesis. We have hematopoiesis for our red blood cells. We call that erythropoiesis. And then we have hematopoiesis for our white blood cells, and we call that leukopoiesis. About 2.5 million red blood cells are produced every second in the bone marrow, and it's going to take about 15 days or so for one red blood cell to fully mature and develop. Erythroblasts are immature red blood cells that still have a nucleus, they still have organelles. And erythrocytes are mature anucleate red blood cells that circulate in the bloodstream and carry oxygen. Now, nutrients that are going to be essential for the synthesis of red blood cells are going to include iron, vitamin B12, and folic acid. So this illustration shows you the pathway from the initial stem cell, the hemocytoblast, and how it differentiates into different cell types before it sheds its nucleus and organelles to become a mature erythrocyte. So we start as a hemocytoblast, which will differentiate into a proerythroblast, which will differentiate into erythroblast. That cell undergoes differentiation and develops into a normoblast. And then the nucleus is expelled, and we have what is called a reticulocyte. Okay. And once the nucleus has been expelled, once the mitochondria has been expelled, we have what will be released into the blood as a mature red blood cell that we will refer to as an erythrocyte. Production of red blood cells is going to be controlled by a hormone called erythropoietin. Erythropoietin is a hormone that is formed in the kidneys, and it's released in response to stimuli such as tissue hypoxia, which basically just means lack of oxygen to the tissue, and or a decrease in the concentration of red blood cells. Okay, so that hormone will be released and it will start the formation of red blood cells. Okay. Now, on the flip side, if we have an increase in red blood cells, we would expect to maintain the homeostatic balance within the body that the kidneys and or liver would stop secreting erythropoietin, okay? Because if not, we could have a situation where we have polycythemia, where we just have an abundance of red blood cells, which makes the blood much thicker, so it increases the blood viscosity, and could lead to some issues with efficient gas transport. So here is an illustration of the negative feedback that occurs when we are in need of more red blood cells. The stimulus is hypoxia, and that could be due to a decrease in the number of red blood cells. It could be due to the decrease in the amount of hemoglobin or just availability of oxygen. This is going to cause the kidneys to release erythropoietin. Erythropoietin is going to stimulate and target the red bone marrow so that we can initiate the production of red blood cells. And then erythropoietin is going to cause an increase in erythropoiesis, which is going to increase the amount of red blood cells, bringing us back to normal, tipping that balance back to what is needed by the body. As a result, we have an increase in the amount of oxygen that can be delivered to the tissues. So all in all, at this point, the body is happy as long as the tissues are receiving the oxygen that it needs to receive and picking up the carbon dioxide that it needs to release. Worn out and damaged red blood cells are destroyed by the liver and the spleen. Heme is going to be broken down into iron and biliverdin. The iron is going to be recycled to be used again, while the biliverdin is going to be converted into bilirubin. Now, biliverdin and bilirubin are going to be secreted in the bowel and then later excreted by the body. All right, so we're at the end of our first video. These are your practice questions. If you have any questions about the answers to these questions, please let me know. But that's it.